Hello everyone. So today we are going to see what are lipids, the introduction to it, the detailed classification, the food sources, the metabolism of lipid and the functions and the disease associated with it. Can be defined as a chemical diverse group of organic compounds which are insoluble in water but soluble in organic solvents like alcohol, ether. The term lipid it is derived from the lipos, which in Greek means fat. Lipid is associated with the storage form of energy and also plays a major role in cellular infrastructure and also the biochemical functions. Now let's discuss how we can classify the lipids. They can be broadly classified into the following classes like simple, compound, derived, miscellaneous and the neutral. So the lipids are classified into following groups, that is simple lipid, compound, the derived and the miscellaneous. Simple lipid is the fatty acid plus alcohol and the examples is talc and the wax. The compound lipid consists of fatty acid, alcohol and also the additional group. This essential example phospholipid, glycolipids. The derived lipids is of hydrolysis of simple or compound lipids. Examples are fatty acid and the steroids. The miscellaneous lipids for the characteristics of lipid. Example is squalene or carotenoids. So let's discuss what are simple lipids. They are the esters of fatty acid with alcohol. They are further classified into two groups. Basically, the neutral fatty and oils and the second that is waxes. Neutral fatty and oil are the esters of fatty acid with glycerol which make this class. They are liquids are solid at room temperature, whereas the oil referred to the lipids are in liquid state at room temperature. The waxes they are made up of entry of fatty acids having long carbon chain with alcohol other than glycerol. The example for that is acetyl alcohol. So now let's discuss the compound or also called as complex lipids. They are basically the esters of fatty acid with alcohol and some additional groups like carbohydrate, protein or the nitrogen base. They are subdivided into phospholipid. Basically, they are lipid containing fatty acid and alcohol esters with the phosphoric acid and sometimes the nitrogen base or amino group. They are further classified into two groups, glycerophospholipid and a sapinophospholipid. This glycerophospholipid, the example is lecithin, and the sapinophospholipid is example is sphingomycelium. Glycolipids, these are the fatty acid and the carbohydrate and the nitrogen base containing lipid. Example is gangliosides. The lipoproteins, they are present larger molecule complex of lipids with proteins. And the last one that is the other complex lipid. These are the esters of fatty acid with alcohol basically containing the additional group like carbohydrate, protein, the nitrogen base and sometimes phosphate. The next lipid is the derived lipid which represent the derivatives obtained from hydrolysis of group 1 and group 2 and having the properties of lipid. The example contains like glycerol, mono and diacylglycerol, lipid-soluble vitamins and also the steroid hormones. The next lipid is the miscellaneous lipid which consists of large number of compounds showing characteristics of lipid. Examples are squalene, carotenoids and hydrocarbons such as phoneticosino and the terpenes. The last lipid is the neutral lipid. They are called neutral as they are uncharged. They are monoacyl glycerol, diacylglycerol and the triacylglycerol. These neutral lipids are commonly found in the cells as a storage fat and the oil. The reason behind they are neutral is that cellular level they have no charge group. Now let's see what are the food sources through which lipid is obtained. The examples are bit fat, the poultry skin which consists of chicken and turkey, heavy cream in the form of fresh milk, butter, soft cheese and the bacon. These are the various food sources through which lipid is obtained. Now let's discuss the lipid metabolism. The first stage consists of the lipid digestion, in which the process of breaking the triglycerides down into smaller monoglyceride units with the help of lipase enzymes. The digestion of fats begins in the mouth by chemical digestion by using the enzyme that is lingual lipase. The majority of lipid digestion and the absorption occurs once the fat reaches the small intestine. The next stage of the lipid metabolism is the lipid absorption, where absorption of fat takes place. The short chain of fatty acids can be absorbed in the stomach, while most of the absorption of occurs only in small intestine. Once the triglyceride is broken into individual fatty acid and the glycerol, along with the cholesterol, they aggregate to form the micelle. The next stage of the lipid metabolism is the lipid transportation. Due to the hydrophobic nature of the membrane, the triglyceride, the lipids, and the cholesterol required a special transport protein known as the lipoproteins. The amphipathic structure of the lipoproteins, along with the triglyceride and the cholesterol, are transported to the blood. The last step in the lipid metabolism is the lipid storage. It can be stored in white adipose tissue as triglyceride. In human young adult, the mass of triglyceride stored represents 10 to 20 gram. The triglycerides form a backbone of glycerol with 3 fatty acids. This slide represents the various steps and the tissues included in the lipid metabolism. The major tissues are the muscles and the liver. The subcellular site is mitochondria. The precursor or the substrate is the acyl-CoA. The end product is the acetyl-CoA. The coenzyme requirement is FAD and NAD+. The pathway increase is in the starvation. And the inhibitor is malonyl-CoA, that is inhibitor keratin acyltransferase. The slide represents the summary of lipid metabolism. We can see how lipid is metabolized through a chart. Now let's see the functions of lipids. The first function is the constituent of cell membrane and the regulate membrane permeability. The second function, it is a source of fat-soluble vitamin. The phospholipids in the mitochondria are responsible for transport of electron transport chain components. Accumulation of fat in liver is prevented by phospholipids. Now let's discuss the various disorders of the lipid metabolism, which includes obesity, hypercholesteremia, fatty liver, and the atherosclerosis. The first disorder is the hypercholesteremia. It is the high level of cholesterol present in the blood. It is approximately more than 200 mg per dl. The condition is said to be in hypercholesteremia. The people with hypercholesteremia have high risk of coronary artery disease and disorders like diabetes mellitus in the case of hyperthyroidism, nephrotic syndrome. It can be controlled by taking proper dietary intake of polysaturated fatty acid that reduce the plasma cholesterol level. The next disorder is the atherosclerosis, in which the artery narrows due to formation of plaque, which is made up of fat, cholesterol, and other substance. It begins with the damage of epithelium and also caused by high blood pressure, smoking, and high cholesterol. It leads to development of some disease like diabetes mellitus, hyperthyroidism, etc. The next disorder is the fatty liver, in which excessive fat deposit in the liver and also the deposition of toxic material. It is also known as hepatic steatosis. Weight of liver increases about 10% than of normal weight. The common causes include alcoholism, heavy drinking, obesity, hyperlemia, and side effects of some medicines like aspirin, steroids, and tetracycline. It is a medical condition, the obesity, where excess body fat is accumulated, 20% excess fat deposition in the body than the normal body weight in the body that causes an adverse effect on health. It is defined as the BMI, that is the body mass index, and evaluated in terms of fat distribution via the waist-hip ratio and the total cardiovascular risk factor. Thanks for watching our video.